Okay, let's get started. Hi everyone, welcome to today's Nakanoan webinar. Just before we get started, I would like to mention the housekeeping items. The presentation slides are available to download at the top of this GoToWebinar control panel. It's called Materials. The presentation is available as a PDF. And also there will be time for question and answers. If you can log your questions in the question box and we will address them at the end of the presentation. And I will hand it over to Peg to start the presentation. Thank you, Peg.
Thank you, Sharon. So today I'll be talking about restoring wetlands in New York City, our lessons learned in urban habitat restoration and sustainability. A little bit about me. I'm a professional wetland scientist and a professional engineer, which is very helpful in the wetland restoration world to have both the science and engineer skills. I have over 30 years of experience in wetland restoration. 25 years of those at WSP, where I'm in the water group. I've restored and created over 2,000 acres of wetlands in Pennsylvania, New Jersey, New York, various states, and also Peru and Egypt. While I'll be giving this presentation, I just want to acknowledge the multiple WSP staff and our subcontractors who have worked on this project and supported it for more than a decade. A variety of engineers, scientists, toxicologists, archaeologists have been involved in moving this project forward. And also we have several sub-consulting firms, women-owned businesses and minority-owned businesses and laboratories that have been supporting the project. Definitely takes a big team. So my plan today is just to give you an overview of the project, discuss some of our key challenges and innovations, discuss some of the lessons learned, and then answer any questions. And I just want to note that all of the photos that I have in here are of the site, mostly recent photos. This is a photo of our biologist, I believe Tom Shinsky, doing some sampling just a few weeks ago. So the project name is the Sawmill Creek Wetland Mitigation Bank. It's located in the northwest corner of Staten Island. I know that we have, I think, an international audience, so I just put up a map of the United States here. New York is up in the northeast corner, and then this is showing Staten Island. The city of Manhattan is here. Statue of Liberty's around here, and New Jersey's over here. So, but this project is on Staten Island within New York City. And it's established by our client, by the New York City Economic Development Corporation. New York City EDC is a public corporation that works to strengthen the city's economy, grow innovative sectors, and deliver sustainable infrastructure. And I want to thank them for working with us and engaging us on this project because it's been a very rewarding, challenging, but rewarding. So Sawmill Creek, the site was previously comprised of filled and degraded urban wetlands. You know, back 100 or 200 years ago, it was all salt marsh, and then it was filled. And we have a history of treating our wetlands and marshes very badly. So it was very degraded. And then there's also some forested areas that were adjacent to the site. So the restoration goals were to remove the urban fill, improve the tidal hydrology, so you have tidal waters coming in and out to support native salt marsh species, control invasive plant species, and increase fish and wildlife habitat. WSP became involved with the project in 2012. This photo on the left shows you sort of what conditions were like then. This is historically a marsh, but it had all been filled in and there was a lot of debris. It took a bit of time, but we got to construction in 2018. Earthwork was conducted in 2018. The site was planted in 2019, and then we've been monitoring it ever since to determine if it's successfully developing as a salt marsh and meeting all the requirements that it was set to meet. So we actually have a nice little video of the project, which we'll share later and you can have access to. This is just an excerpt from the video. But to achieve these objectives of restoring the salt marsh and providing ecological benefits, there's a long and complex process involved. And this photo is actually showing after the site had been constructed, prior to it being planted, we had to take sediment samples to make sure that it was clean prior to planting. So this is a channel that was constructed. So just keep this in mind. This is what it looked like during construction. And this is just an example of how the site was restored. And this is the same location where people were standing in that photo. So the people who were taking samples were in this channel here that we constructed. So the photo on the left is what the site looked like prior to restoration. You can see all the debris that was on the site, and there's this trailer that's adjacent to the site on another property, which just helps you give perspective. So on the left is what it looked like prior to restoration, and after restoration, these are the channels that we designed and that were constructed to bring tidal waters in and out of the site. And then this is that same view um, after it's restored, where we created the channel. That dilapidated blue trailer is back here. Um, and the photo previously where people were sampling was taken right here. So I'm just going to go over some of the challenges that we faced during this project because I think they are helpful and applicable to other urban restoration projects. Um, the main challenges are the design, the fact that we had to work with multiple stakeholders. There are also archaeological resources, but really the biggest was the contamination. Also, there's debris dumping on the site, and then there's a concern from the regulatory agencies about recontamination. I'm going to be talking about that a bit. So in terms of the design, this is a salt marsh restoration design. WSP has a fairly standard, successful approach to coming up with that design. We generally look at where native salt marsh species are growing well, which we call biobenchmarks. We use reference wetlands to find out what elevations they're doing well. We also monitor the tidal hydrology, get several months of data, so we have a good understanding of where mean high water is, mean high water. We analyze reference streams and we use that to help design our tidal streams. And then based on all that information, we set the design elevations to support the tidal inundation. Both the water needs to come in and also come out. It's just important for a salt marsh that it drains is that it gets flooded twice a day in the low marsh and frequently in the high marsh. And then we accommodate for sea level rise and develop a native planting plan. So our planting plan for this site is here. You can see the elevations that we set for the low marsh is 1.5 to 2.5 feet. This is all an NGVD datum. We had about 5 acres of low marsh, and really the only plant that grows in the low marsh is the Spartina alterniflora. We had high marsh with a variety of plants, shrub marsh, and then upland buffer. So really the design was fairly straightforward. We were done with the design in about a year. The challenging part was getting all of our approvals. And again, this is just an overview of the various habitat types that we created. The areas in green were upland areas that were no longer wetland, so we restored those back to wetland. The dark blue areas were very degraded wetlands, and we restored those back to native salt marsh species. And then this lighter blue area was some pretty nice salt marsh that was there, but it had some invasives and a lot of debris, and we enhanced that. And then surrounding the site is upland buffer that we also removed debris and invasives. Um, So like I said, the design was fairly straightforward, but the reason it took us 4 years after the design was complete to get to construction is that there were a lot of project stakeholders. We had to get a lot of permits. We had to coordinate with a lot of different people. It's in New York City. The land is owned by New York City Parks, and they were a strong partner throughout the process. But we had to negotiate with all these variety of agencies. So I would say our approach here is frequent and open communication. That was really required, but it certainly took several years to work with all of these agencies. In terms of archaeological resources, our archaeologists coordinate with the State Historic Preservation Office and other regulatory agencies, and then our archaeologists were on site during construction in the sensitive areas. The main concern in the urban area is contamination. The site had a lot of debris in it, had our pre-construction sampling indicated that there were contaminants on the site that were above the ecological criteria, not for the most part not above human health criteria, but it was a concern to ecological health. So to deal with that, our design removed most of that fill. We removed over 40,000 cubic yards of material. We also excavated channels and the contaminated areas, and in some places we were removing about 10 feet of fill. Um, and then, as I mentioned, once we had the site all graded, we had to collect samples to make sure it actually was clean, that we removed the contamination. And all those sediments and samples were collected by WSP, and then we compared it to the New York State sediment guidance values to be protective of aquatic wildlife. So we needed to be, our samples needed to be below those standards. And if they weren't, then we placed clean sand. And there are some areas where we weren't, and we over excavated and placed clean sand so that the site was all clean prior to planting. It was also very helpful to have an experienced construction team. WSU was the designer, and we were involved during construction. Construction management was provided by Lero Engineers. Galvin Brothers was a construction contractor. And then the marsh plants came from native nurseries like Pinelands and Greenbelt. Um, Once the site was restored, we did need to make sure that midnight dumping was curtailed, which again is an issue in urban areas. So we did put a very tall fence around the site, about 10 feet tall, and installed signs, and we have to monitor it frequently. And the main thing I wanted to talk about, the main thing that's most challenging, I think, or the biggest question for urban restoration in our area is once you restore this site, once you clean it up and you're providing something that's not contaminated, is there a possibility that it could get recontaminated again? You know, this is a tidal wetland. The tide comes in and out twice a day. The tide has sediment in it and deposits it on the wetland, and that's all part of a normal process. That's one of the ways that marshes keep up with sea level rise. But since we are in the New York, New Jersey harbor area where there is a lot of historic contamination, there's a lot of Superfund sites in the area, there is a concern by the regulatory agencies that the site could get recontaminated with sediment coming in on the tides. So the regulatory agencies devised a plan for us to implement, which was to study how the marsh was doing after restoration. So the items on the left, the annual monitoring and maintenance, this is fairly typical maintenance monitoring that's required of a restored wetland, and we conducted all of this work. And the items on the right are requirements in our permit that was specifically addressed at assessing whether or not contamination, recontamination was occurring on the site. So I'm just going to talk about the first part first, the annual monitoring. And so far, our annual monitoring program, we've done 4 years of monitoring. This is our 5th year. We actually have some of our scientists and New York Parks out today continuing this monitoring. This will be our last year of detailed monitoring. And we found that it's 88% covered by native vegetation, which exceeds the criteria that was set in the permit. There's very little invasive species, which is well below the 10% limit. We have 2%, and it's well below the 2%, the 10%. So we're doing very well in terms of vegetation and in terms of providing a native salt marsh habitat. We're seeing smooth cordgrass, salt meadow cordgrass, other salt grasses growing in the area. We have, in terms of birds, we've seen over 33 species, including osprey and salt marsh sparrow. We actually have people out today who again saw salt marsh sparrow on the site, which is amazing because that's a species that's widely considered to be one of the most rapidly declining avian species in the area. So to see it on our restored wetland is very rewarding. So this meeting all of its criteria, and it's very a successful vegetative and wildlife habitat. So in terms of contamination, recontamination, the agencies require that we look at the 3 wetland disturbance areas. That's where we did earthwork. So we call the one on the north Edward Curry, the one in the center we call Chelsea, and the one to the south we call Bloomfield. They're named after the roads. This is an urban area. And then we also compared it to a reference area, which is the salt marsh that was undisturbed. The program was developed by the Fish and Wildlife Service, and WSP has been implementing the program on behalf of EDC at these various locations. And this is our last year of monitoring. So the idea is if sediment is coming back on the site, is it recontaminating? Well, not if, when sediment comes back on the site, is it recontaminating the site? And even if it is recontaminating the site, is that sediment then getting into the biota and going up the food chain? Is it causing a problem? So that's really the goal of this monitoring. So for the sediment sampling, we're only looking at the top 1 centimeter of sediment. So that's basically just to take samples of what's being deposited on the site. So we're trying to determine if the sediment that's being deposited by the tide is contaminated and causing a problem. And then we compare that to the data we collected at baseline right after it was restored and see if there's any change. And we're doing that at the wetland disturbance areas. And we're using a very detailed compositing scheme called incremental sampling methodology. And then we're analyzing the samples for PCBs, for dioxins and furans, pesticides, metals, mercury, total organic content, carbon, solids, pH, and grain size. And again, these are all requirements in our permit from the U.S. Fish and Wildlife Service. This is just a graphic of the very detailed sampling that occurs. It's very labor-intensive field work and also a ton of samples to be analyzed. But like I said, we have the 3 decision units. Well, for each of the wetland disturbance areas, we have 3 decision units shown here in light green, which is the high marsh. The darker green is intertidal marsh, and then the blue is the waterway. And in each of those locations, we take 20 samples, set areas, and we composite those. And we actually do that 3 times. So we have 3 composite samples for the high marsh, for the intertidal, and the waterway. So it's very representative of the whole site of those areas. So it's a total of 180 samples that we collect in order to come up with these composites. So the whole goal of this is to collect a representative sample by generating a composite sample across the grid. And I don't. And for the biota tissue sampling is, again, to look at, even if we found that if there was contamination on the site from the sediment, is it making it up the food chain? So we're analyzing species that spend a lot of time in the sediment. We're analyzing mummy chog fish, fiddler crabs, amphipods, spiders, 2 kinds of spiders. So we take 5 composite samples from the 4 areas, which are the Edward Curry, Chelsea, and Bloomfield, where we did wetland disturbance, and then also the reference area. And we're analyzing those for PCBs, dioxins, furans, pesticides, metals, and mercury. So the goal is to assess if these contaminants are accumulating in their tissue, because if it's in the tissue of these organisms, which get eaten by birds and go up the food chain, it would be a problem. So that's what we're trying to assess. That's the point of the sampling. I'm going to go over some of our results. We have a lot of data, but I'm just going to give you a little bit of information. In terms of the collecting the biota, we were able to collect all the fish, the mummy chogs, the fiddler crabs. The amphipods are teeny, so we were able to collect a lot of them, but not all that we would have liked. And then again with the spiders, they're very small mass. And we are working with an analytical laboratory that can sample a very small mass of tissue, of spider tissue. So to go over our results, just as an example, this is some results of the sediment monitoring. I'm going to focus on mercury. These samples here on the left in the gray were the concentrations of mercury that we saw on the site prior to restoration. So you can see we had fairly high mercury levels prior to restoration. We removed that contaminated sediment or replaced clean sand, and at the start of the planting, mercury is down to 0. And then the samples that we've been collecting after restoration are shown here. The high marsh are in green. If it was not detected, we have a green X. The intertidal marsh is shown in this orange color, and the waterway is in blue. So we're basically seeing, we're not seeing a huge increase in mercury over time. This is indicating that we're not really seeing any recontamination of mercury. It is slightly higher than what was there when we just finished construction, but it's nowhere near what it was like prior to construction, and it's not really showing any kind of trend upward. So that's good. It does not appear to be contaminated, and we're seeing this really for all of the areas and all of the parameters, but I just want to show you this one as an example. And then again, as an example for the biota data, this is for mercury. This is for the fiddler crab, which spends a lot of time in the mud. The results prior to construction are here on the left, and then after construction are here on the right. And then the blue lines that it's between are the reference area. So this is showing that after construction at the Edward Curry, which is the northern part of the site, we're not seeing any kind of increase in mercury in the crabs, and it's similar to what's in the reference area. At Chelsea, which is the center part of the site, the first year of data we had was a little concerning because it was above the reference site, but since then we've seen it go down. Again, we're not seeing any kind of upward trend, and it's very similar to the reference site. And that's also true for the fiddler crab that have been collected at the southern part of the site, the Bloomfield site. So just to go over the summary of our results on these recontamination studies, and again, I'm going to focus on mercury. This is a lot of data, but what we're seeing is that the sediment composites collected in 2021, 2022, and 2023 suggests that the sediment composition has not changed since construction. So we're not seeing that the sediments on the site are being recontaminated, which is very good news. And then for the biota samples, I'm just focusing on year 3 and 4 since we had that anomaly in one area. But it's showing that the tissues were comparable to the range of mercury in the reference area and either less than or comparable to what was there before. So we're not seeing an increase in contamination in the biota either, which is good news for urban restoration. This is our last year of sampling this year, so we'll finalize this report this year. But so far the data we have are indicating that the site is not getting recontaminated or posing a risk to wildlife. So our lessons learned is that the wildlife utilizing the restored site are exposed to much lower concentrations after restoration. And the sediment and biota tissue results are comparable to baseline restoration reference site conditions, suggesting the wetlands not been recontaminated. And we're hoping this information will help support other restoration projects in this urban area and eliminate the uncertainty regarding recontamination. So those are our lessons learned generally about recontamination. Overall, the key takeaways on the project is that the New York City EDC expects the Sawmill Creek Pilot Wetland Mitigation Bank to facilitate larger wetland restoration projects in the city, particularly in their ecologically sensitive coastal areas, also allowing to direct more public and private funds for restoration of damaged ecosystems. Improving the city's resiliency to climate-related disturbance, applying lessons learned to restoration of other degraded habitats, and the project serves as a model to future wetland restoration projects within often overlooked and marginal urban areas. And I want to thank everyone for their participation, and with that, I'd be happy to answer any questions.
Thank you, Peg. Fantastic presentation. Just mentioning the housekeeping items again, the presentation slides are available to download at the top right. There is a material tab, and also please continue to log your questions in the question box. We have some time for a few questions. I will start with the first question. How would you prevent contamination coming in from tidal flows? And what would you do if you do find increased contaminants in sediments?
Yeah, that's a really good question. I'm not sure how you could prevent contamination coming in from tidal flows. I mean, um, there was a concern also on this project that there had been historic oil spills in the area nearby. So we did actually put an oil boom in there that could be deployed quickly. So if there were an oil spill, we could deploy the that boom and it would keep the oil from coming in. I think the way you prevent contamination is just continue to have the area cleaned up. There are a lot of Superfund sites in the area that are in the process of being cleaned up. Once those are cleaned up, we'll continue to see improved water quality and sediments in the area. But for the questions for what would we do if we find increased sediments, contaminants in the sediments, there wasn't that was it's a good question. This was basically a kind of a research study to get the information. So there was nothing set out about what would happen if we found it. It was to get the information. And then I think at that point we would have had to figure out what to do. So luckily we didn't find increased contaminants in the sediments and we didn't have to deal with that.
Thank you. The next question is about the monitoring program. How was the duration of the monitoring program determined, and are there any plans for future sampling in the future?
The duration was determined by the regulatory agencies that issued our permits for this project. It's fairly standard to monitor the site for 5 years, to monitor restored wetland for 5 years. So that's how the 5 years was set. But once this 5-year monitoring period is over, there is a long-term steward in place. Like New York City Parks owns the site, and they'll be taking over monitoring of the site for the long term, but it will be much less intensive. It won't be any biota or sediment sampling. They'll be amazingly keeping an eye on the site to make sure it continues to function successfully as a tidal marsh.
Thank you. There's a question here. What was done with the removed contaminate material?
The material that we removed from the site, like I said, was not generally above human health standards contamination. It was just above the ecological criteria, which is important since this is an ecological habitat. But for the most part, that material was taken to a landfill. There was one at the heart of the site that had very high levels of PCBs. Those had to be treated separately. They were considered toxic, and those had to be shipped off to a facility that handles high levels of PCBs, which I think was in Michigan.
Fantastic. Thank you, Peg, for a fantastic presentation. Apologies, we have a lot more questions coming that came through. These will be answered by the presenter later on. So again, thank you everyone for joining, and thank you, Peg, for a fantastic presentation. We look forward to seeing you in our upcoming session. Thanks, everyone.
Thank you.